All right, let's get started. So last day, you guys heard a little bit about protein language models, and today we're going to dive into models based on DNA. We'll continue to talk about transformers, but then one of the main types of models that's used with DNA in particular is convolutional neural nets. So we'll spend some time today talking about convolutional neural nets as well. All right. So the first problem that I want to talk about is it's a tricky one. And so this is the process of going from a pre-RNA to like a fully spliced RNA. And the signals involved in RNA splicing, um, they're maybe not as clear as some of the other genomic signals that we have. So it's a difficult problem. And it's important functionally because, we can have a gene can tend to be very long with very small parts of it actually involved in the coding sequence. And then the function of the gene can change when there's multiple different options for how to splice it. So figuring out what's a real option that might actually be seen, let's say in RNA-seq data, versus just a theoretical possibility is an important but challenging problem. So I say it's a hard problem because the splice sites, like the donor site, is basically like two nucleotides, GT, and the acceptor site is two nucleotides, and there's some very weakly conserved additional signals that are involved in making a biologically active site. So for quite a while, this was a problem that prediction tools perform very poorly on. I want to dive into one particular tool that I think when it came out, it really kind of set the standard for prediction, and that's SpliceAI. So SpliceAI is it's based on convolutional neural nets, and so there's an input DNA sequence that goes in. And then it goes into this convolutional layer. And I want to dive into that just a little bit, okay? So to do that, like I want to talk about what exactly is a convolution. So convolution is when we have basically two functions, and they operate together to form a third function. And I want to illustrate it with some of my research. This is probably the the only time I've really used convolutions myself. And this was based on this paper that we published like really early on in the COVID pandemic. So one of the things that I had done in my lab in the past is wastewater surveillance and trying to estimate the incidence and or prevalence of different diseases by looking at how much shedding of those diseases, either like bacterial or viral, there is in wastewater. So when COVID happened, my lab kind of switched from mostly being interested in salmonella and dengue and other pathogens to really just focusing on how much COVID there was in wastewater. In one of the first papers that we had out, and this was the first time that anyone had like actually sequenced COVID in wastewater to really prove it was there, was this paper. And what we found was based on how much shedding of the COVID virus we found from folks who were hospitalized for COVID, which is where we got all the clinical samples. If we just took the amount of viral particle that they were shedding, and then we looked at, how much virus there was in the wastewater and, divided the one by the other, we got enormous amounts of people being infected. And this was really important because if it was true, then it probably meant that COVID was, at least at the beginning, a lot less virulent than we imagined, because, we based on those numbers, we were predicting that very early on in the pandemic, like 30-40% of the Boston area like actually had COVID and was just asymptomatic, right? So became really important to figure out is this true or not? So I want you to think about kind of the following problem, which is how we thought about it. So let's say there's a new virus that appears in Boston, and each day, every infected individual excretes 100 like viral particles or 100 units, maybe by some PCR assay into wastewater. Each new infection lasts for 5 days. Okay. And if we say that, this is the time course on day one, there's 10 people that are infected. Day two, there's 25. Day three, there's 150, then 50, then 25, and then no new infections. How much virus do we expect to see in the wastewater on any given day? I want you guys just take 1 minute and plug through a few of those days. Okay. Put in some numbers for days 1, 2, 3, 4. All right, so day number one, how much virus do we expect to see? 1,000? Yeah. What about day number two? Yeah, right. Yeah. So we have 2,500 from the people who are newly infected on day two, but we still have 1,000 from the people who are infected on day one, right? And if we go to the next day, then we've got 15,000 from the people who were infected on the third day, plus 2,500 from the people who were infected on day two, plus another 1,000 from the people who were infected on day one, right? And so then we're gonna continue this math, and then when we get to day five, now like we're not counting the people from day one anymore, and when we get to the next day, then we're not counting the people from day two anymore, and so it's gonna fall off, right? But there's this sort of lag time. And now we can think about like how would you solve that problem if the infected individuals gave off 200 units of virus on the first day, but then only 100 units on the next 4 days, right? Like you could go through and you could solve that problem. And this is basically what we did is we worked backwards, and so we looked at we looked at the new cases, and we tried to estimate, what is this function that describes how much virus people are excreting, and eventually what we were able to do, which kind of solved this paradox that we found, where there was way too much virus for the number of reported cases. what we found is that the shedding function that we observed, and most of the people we observed, they had had the virus for a long time, okay, and they were already hospitalized, and so when we measured the amount of virus that they were excreting in fecal matter, it was actually much lower than it was on the day or two following infection when they were probably asymptomatic, right? And so, it's hard to collect those samples. So, eventually, we did get some data, and it supported our like inference of this true shedding function. But this whole process of figuring out, how much virus is there on any particular day. It's basically going from the number of new infection cases. So this function I of t, combining it with the shedding function, which is on any given day, how much is each person shedding? And we're combining those through a convolution, right? That math that you were doing to solve that problem is basically a convolution. We're convolving the number of new infections with the shedding per patient, okay, to get the viral concentrations. And so what we did in a follow-up paper is we looked at the viral concentrations and the new infections, and then we sort of reverse engineered the shedding function, and it actually was confirmed first in mice and later in clinical samples. Okay. So that's the idea of a convolution, and a convolution, we can think of it in terms of mathematical functions, which are continuous, or we can talk about it in like discrete units, like, how much do we expect to see on any particular day? Okay. And I'm going to just define a few terms. So my input, that's going to be a sequence, could be a DNA sequence, or number of viral particles, something like that. My kernel, that's kind of my viral shedding function. That's telling me, like, what am I going to do to this input sequence? That's kind of what's performing the convolution. The output of performing a convolution, I'm going to refer to as a feature map. And then I can talk about the width of my convolution. So in the case I just described, the width was 5, right? The convolution only acted over 5 days in a row. And then when we're using these convolutions as sort of a tool to analyze deep in the context of deep learning, we'll also talk about things like the stride, which is sort of like how we apply the kernel. And I'll just briefly mention a few issues around how to do this practically, like padding the original data with zeros. Okay. So I want to walk through an example. If you look at the URL here, I just want to point this out. So I'm taking these slides from a really nice course on deep learning that was actually developed by undergrads at Duke. And so I encourage you guys to go back and check those out. There's a lot of really good material on there that kind of walks through a lot of the concepts that we've talked about with nice illustrations, things like that. So now let's talk about convolution in 2 dimensions. And this is one of the most common uses of convolution. So imagine that we have an image of like a handwritten number 3. Okay. And so you can see how it's shaded, and then I put numerical values that correspond to the shading. So I'm going to take this image and I'm going to convolve it with the kernel that you see in the middle. Okay. And so the way that I'm going to do that is, first I'm going to put the kernel in the upper left-hand corner. and I'm just gonna pairwise like multiply all those corresponding values, right? So you can see in the kernel, there's a -1 down the left-hand side. There's a 0 in the middle column, and on the right there's a +1. So I'm going to take all the values in the input image and multiply them element-wise by that kernel. And so then in the convolved image, once I add all those up, I get the number 60, right? So I'm just going to put that down for that upper left-hand spot in my convolved image. If I move it over 1 and do the same math, then I get the number 10. So I'm just going to like put that in. And if I just raster across the entire image, then I get this new convolved image, right? And if I look at that image, now I can start to see some patterns, right? Like where is most of the signal here? Where's the highest magnitude in signal? Well, it's kind of right down the spine of that number 3. And this type of kernel is really good at like identifying vertical lines. Okay. And so different kernels are going to perform different functions. And oftentimes in neural nets that do image analysis, in some of those first layers, we see kernels develop that that look for vertical lines or horizontal lines or diagonal lines or gradients or simple components like this. Okay. I want to talk about a couple more things that you'll see and are worth mentioning if you're going to make your own convolutional neural nets. One is stride. So stride is a way to kind of reduce the amount of computation that you're doing and reducing the total size of the image that comes out. So you could imagine doing the same thing, but then instead of stepping through every square of my handwritten 3, I can just I can take 2 steps because my kernel is length 3 and it's still going to see all the data, right? So I can do that, and then I get a convolved image for the same handwritten 3, but with stride 2, and it's much smaller than my convolved image for stride 1. However, if you look at the highlighted areas, it's identical to stride 1, right? We're just taking a subset of all of the values in that matrix. Okay. Now, the one thing to remember when you're actually making convolutional layers and using them is, if I have a 10 by 10 image and, I apply a kernel of size 3 to it, how many rows and how many columns can I actually look at? Well, it's fewer than 10, right? If I start in position 1, I can only get to the point where like the I plus 2 value of the kernel is 10, right? So I'm losing space along the edges, right? I'm losing 2 spots. So if I want to have a bunch of convolutional layers one after another, the output is going to keep shrinking down and down. And so that may be a problem. And so what a lot of people do is they pad it so that the output actually equals the input size. And I can figure out what the output size of my convolution is by just this simple formula, which is just what's the input size minus the kernel size plus twice the padding size, because I have padding on both sides, and then I divide by the stride. Okay, and that and add 1, and so that's going to be the size of the output of that convolutional layer. Okay, so like why are we making these convolutional layers? Well, like one way to analyze, let's say, this handwritten digit, and probably like the easiest way to do it would just be to, to take that two by two matrix and just splay it all out in one dimension, right? So if it's like 20 pixels across, then we would just make one vector of 400 values and just, put them into a fully connected neural net. And that's probably fine. And we can train that network to recognize those digits pretty well. But like there's a lot of parameters in that model, right? Because now I've got for every layer, I have 400 neurons that are connected to 400 neurons in the next layer, so it's 400 squared. Whereas we know that it's only the pixels that are most important to interact with each other are the ones that are close together in 2D space. So when I splay it out all in one dimension, I've lost that. So with convolutional neural nets, the number of parameters in my model is really just the size of that kernel, right? And so it's not going to be like 400 by 400. I can control it by controlling the kernel size. And so that kernel is probably going to be relatively small. And what will typically happen is I'll have like multiple channels. And so, I'll have one channel that might be my image, just looking at putting it through the vertical line detector, and then another channel taking the same image, putting it through the horizontal line detector, and all of these different operations that are coming out of my convolution kernels. So it's pretty efficient, and it preserves the two-dimensional nature of the input, and requires a lot fewer parameters. And it requires fewer parameters because well, one of the ways to minimize the number of parameters is to take that larger image and then collapse it into a smaller version. So I get I get those lower res features, and so if I do a convolution on the full resolution image, then my kernel is only looking at local interactions. But if I can condense it into something that's lower res, and then I have convolutional layers that act on that, then I'm starting to pull in longer range interactions. And so that's one way to get the longer range interactions in. And so one of the ways to reduce the size of the output is this max pooling operation. And it's kind of exactly how it sounds. So if I want to take this 4x4 matrix and make it a 2x2 matrix, then I'm just going to divide it. So I have subsets that, and 2x2 groups. And within each group, I'm just going to take the max. And so I can do max pooling, which is probably the most common, but you can also do average pooling. And it works as you might expect intuitively. So convolution has become fairly important in the field of machine learning. And it really kind of took off in 1989 when Jan LeCun developed a convolutional neural net to recognize those handwritten digits. And kind of one of the big advances there was it was one of the first times, especially for a CNN, where backpropagation was used to figure out like what those parameters should be. So like what should those numbers in the kernel of the convolution be? So those are the parameters of the model. And I showed you the vertical line detector. These things kind of naturally emerge in these kernels when we just allow the model to figure out what the kernel should be, to figure out what those parameters are. So in 1989, Jan LeCun used backpropagation to do that. Worked really well. Now, virtually like all the deep learning models we use are training parameters by backpropagation. In 2012, Alex Krzyzewski trained like a much larger convolutional neural net, and this is when we're really starting to get into deep learning. I think this model had like 60 million parameters or something like that. It was much larger, I think, than other models in the ImageNet competition, and this CNN that he named AlexNet was like really won by a large margin, and after that, kind of the field of image recognition really moved to using CNNs, and one of the was also one of the first times where training a big model was done on GPUs, and that's also something that's pretty much ubiquitous now in deep learning. So these CNNs have played a big role kind of in the history of deep learning, and even though like a lot of what we're doing is transformers, when we start to talk about DNA sequence models, because there's so much DNA, like the sequences are just so long, it's becomes kind of difficult and expensive to use transformers. So these convolutional neural nets really play an important role. So how do we do a convolution on a DNA sequence? Well, first, we take our sequence, and then we convert it to these one-hot vectors. And so these are one-hot vectors of ACTG. So those are the columns on the matrix that you see. The rows are the positions in the sequence, and they correspond to these one-hot vectors, right? Where if it's A, then only the first position has a 1, and the rest are zeros. And so these are the channels. So our input has 4 channels, and then once we put that into our convolution, it may have more than 4 channels, in any particular layer. And the important thing to realize is generally in a convolutional layer, the input layer might have, let's say 4 channels. The output layer might have 4 channels. Each of those output channels is connected to all of the input channels, okay? And so what this means is if I want to have a kernel that detects the sequence ATG, I can do that because I can have a convolutional layer after my input that's looking for a 1 in the first position, or an A in the first position, a T in the second position, and a G in the third position. So it can kind of mix and match from all of the different layers before it. If we have 4 channels, it's not just performing a convolution on one channel at a time. Each channel in this layer is connected to all the previous channels, right? So it's a very convenient way to represent sequence motifs. So in general, this is what a convolution might look like for a DNA sequence. So we'd have these 1D convolutional layers. They might have many channels. Those channels might, they could be sites that are important for chromatin structure. They could be sites that are, enhancers or repressors. There's a lot of different things that may be encoded in these DNA models, depending on how we train them. Okay, so now let's go back to SpliceAI. So what's going on in it? Like initially, I showed you the 80 nucleotide model. The 80 nucleotide model is pretty simple. It doesn't work. It doesn't work that well. But you know, they show it for comparison, and we can step through that. So we have our input, and our input now is you know, our DNA sequence, 80 nucleotides, you know, splayed out into these one hot vectors. And then that goes into a convolution where, so they represent it with three numbers here. One is just the batch size. So it's just like how many samples we're looking at any particular time. But the important two numbers are the W and D, so the width and the dilation. So the width is the size of our kernel. Remember, it's a 1D kernel, so there's one dimension. And it's saying, you know, like how many positions are we looking at? Just like when we were talking about the viral shedding function, we had a kernel of width 5. And in SpliceAI, they have these kernels of different width, and most of them are width 11. Okay, so they're looking at 11 nucleotides at a time, which is pretty good, right? What is 11 is basically how long it takes the DNA to kind of make one the double helix to make one full 360-degree turn, right? So if you're a part of a protein that's recognizing some sequence of DNA, like 11 is a pretty good number, right? And then you can, you know, you can join those 11 mers together if you want. Okay. And then these convolutional layers are kind of like one after another, and then you can see like within this unit, there's something called a batch normalization, ReLU convolution, and they're kind of stacked on top of each other. So when we talked about transformers, we talked about normalization, we talked about layer norm. Batch norm is taking each activation in our neural net and making sure that, you know, if we put in 32 sequences at a time, the mean and standard deviation of the activations of that neuron are, you know, 0 and 1 respectively, and that just makes these deeper networks a little easier to train. ReLU, again, that's our nonlinearity, so that's our activation function, and then we have our convolution, and you can see this this motif is repeated twice, and then you can see that arrow coming down and joining up again. And so this has got one of those residual or skip connections, and that also helps train these much deeper models. And if we look on the right, we can see the 10K model. We're going to have to move there we go. And move the picture of me. We can. Maybe we can't. Okay. No, that's annoying. But if we look at the 10K model, you know, it basically has it's got the 80 nucleotide model built in, and then you know, then it goes into you know, another block, but now with a larger value for this D parameter, and so D is dilation. And dilation acts, it acts a lot like the stride, but what it does is it takes the kernel and it expands the kernel itself. Okay, and the kernel will just now if you have a dilation of 2, the kernel is going to skip every other residue in the sequence. So if I have a kernel of size 3 and I have a dilation of 2, then it's going to look at one nucleotide, skip one, look at another one, skip one, and look at another one. So if I start with a kernel of size 3, I end up with a kernel of size 5. Okay. And so that's what the dilation is doing. And you can see that in that 10K model, first they have a dilation of 4 and then 10. And then they have a very large kernel of size 41 with a dilation of 26. And so that thing is looking at enormous tracts of DNA, right? And so it's getting a lot of that long range information. And then the output of each of these layers is just added together. So they're all, the really fine grained stuff and the really long range stuff, they're all added together. And the idea is that, if our embedding space is big enough, it can handle a lot of information without kind of overwriting each other. So we can add those together and then at the end, make some prediction. Okay, and in this case, the prediction is it outputs basically 3 numbers, right? One is donor, saying that this site in the DNA is a splice donor. One is acceptor, and then one is like not neither, basically not involved in splicing. And then we take those logits, apply a softmax, and now we get a probability for each point in the DNA, whether it's a splice site or not. So that's basically how SpliceAI works. And it ends up making fairly good predictions. And so you can see kind of in this middle track here, you can see like the true values, the splice donor and acceptor, and then you can also see the SpliceAI predictions. And then here's one of the other leading programs at the time, just to show you kind of like what an advance on the field it was. Okay, so previously you could kind of tell if something was a donor or acceptor, but there were a lot of false positives as well, and it wasn't really obvious. If you wanted to be sure, you'd probably have to get some RNA-seq data and to really know how a particular gene was spliced. But with SpliceAI, like for many genes, you can actually go off the prediction. Okay, so all of that is an example of how we're training these networks to perform specific tasks with DNA. And this is a little bit different maybe than some of the things that we're talking about with the protein language models, where we were taking those models and using them to, predict what's the missing amino acid, right? We're just like training them to generate new plausible sequences. And so we can do that with DNA too. And so that's the idea behind a DNA foundation models. So these are like very large networks trained on like a huge corpus of data. And generally they're trained by looking at a DNA sequence, masking out certain nucleotides or strings of nucleotides like k-mers, and then trying to predict what the masked out sequences were. And in order to do that, these models have to be able to learn a lot of the principles required to compose a DNA sequence. And in order to do that, they need to be trained on massive amounts of genomic data. But the good news is, like we have massive amounts of genomic data. And then these models are very general, right? They're just able to predict what a likely DNA sequence would be, and they're able to do that through the internal representation that they have of the DNA sequence. And the hope is that if we take that internal representation, it has a lot of information in it, right? Like the internal representation for the SpliceAI model, it really mostly probably has information about DNA splicing or RNA splicing, but these DNA models, they probably have tons of information. They probably have information on splicing. They have information on, regulatory sites, enhancers, chromatin. It's all in there. And so the idea is let's train these models and then let's take those internal representations, those internal embeddings that are coming out of those models. And then instead of our training data being just a DNA sequence, our training data is going to be the embeddings from one of these models. And then if we train it to do different tasks, hopefully it performs pretty well because there's a lot of information there. So it's an example of transfer learning where we're training a very large corpus of data on one general task, and then we're gonna leverage that to perform specific tasks. Okay, so I think we talked about ProtBERT in the last lecture, and so this is a transformer encoder for DNA instead of protein sequence, but operates very similar. And so the idea is, if one way to look at sequences we talked briefly about would be these recursive neural nets, and there's a lot of issues with recursive neural nets because they have to pass information like one node at a time, and each node is sort of summarizing all of the information from the entire sequence before it to the next one, and then that node has to add its information and then also figure out what's important from like the entire sequence before it. And it's kind of like this telephone game. So by the time you get to the end of a very long stretch of DNA, there's very little information from the beginning of the sequence there. It's just really hard to preserve that because it's being bottlenecked at each step. Convolutional neural nets are a way to get around that. So you have all the input. So here, you can kind of see on the bottom there's 5 tokens in the sequence, and they're colored according to what that might look like in the embedded space. And the convolutional neural net it's sharing information between adjacent sequences. And then the transformer, the nice thing about the transformer is everybody gets to talk to everybody, and everybody gets to share their information with everybody, right? So you're getting a lot of you're getting a lot more information out of the transformer than either of the other two. So DNABERT is it's a BERT type model trained on DNA. It's trained not on single nucleotides, but on k-mers. So multiple short strings of nucleotides put together. And basically we can walk through sort of how the input goes in and what we see at the end. So if you look on the bottom right, that's like our input sequence. That's tokenized in this case into 3-mers, and then those are embedded into some space that's that is meant to represent the properties of that 3-mer. Once we have that embedding, then we add the positional encoding, like we talked about earlier, and that could be those sinusoidal functions or something else that adds information that says what position was this trimer in the sequence, and so that becomes the input embedding, and then that goes to like multiple of these attention layers. So all of this should be like relatively familiar to you guys, and that's basically the principle underlying DNABERT. And so there's 12 of these attention, self-attention units followed by the feed forward network, and then it and then it outputs, and it's trained on just predicting what the mass sequence was. So so that's how the initial model is trained. And then once we have that model, then we can take that pre-trained model and now we can fine tune it to for some downstream task. And so if you look at at the panel in the bottom left, it's really kind of cool. And so the reason there's like 12 vertical images is because there's 12 different attention layers, and what you're seeing in each layer is like where the attention is going as it makes its way through through the network. And you can see for each of the different attention heads, it's collapsing down to these like two spots, right? So all of the different attention heads are paying a lot of attention to, by the time they get to the last layer of that network, they're paying a lot of attention to these two regions of the DNA string. And it turns out, like in this case that they're showing here, like those are, I think those were two different transcription factor binding sites, right? And so these models that like the attention it's figuring out like what's important in this DNA sequence, and then using then like most of if you were to grab the embeddings that representation that comes out of that final layer, it's going to have a lot of information about those DNA binding sites, right? And that's intuitively sort of the behavior that you want. Okay, so that's DNABERT, nucleotide transformer. They're very similar, but I want to walk through it. So nucleotide transformer is just another transformer trained on large corpus of DNA data. Works about the same. So you can see on the top, that's kind of the path for training. And we start with the DNA sequence. We change it into k-mers. Here it's like 6 nucleotides at a time, so like 6 mers. And then some of those nucleotides are masked. And then we're training it to predict those masked nucleotides. And then once the model is pre-trained at that prediction task, then we can take the same model and freeze most of the parameters in it, and then take the embeddings that come out of that initial model and put them into a new network that's designed to perform some sort of prediction. So in this case, it's like outputs 2 states, like it is a promoter or it's not a promoter, and then we can train that. And while we're training it, most of the parameters that are varying, they're in that network that's basically just using the embeddings that come out of the initial network. So that's kind of on a like nucleotide by nucleotide basis, but we can also make predictions for proteins. So here's an example that they give of if we wanted to know if this particular DNA mutation was deleterious to the protein. So now we're making a protein level prediction, and so we can put the 2 sequences through the initial transformer that was pre-trained, and then in this case they just average all of the residues together to get like one embedding that summarizes the entire sequence, and then they can train on that, right? And they can use that to predict if something's going to be deleterious or not. And in this case, they do 0-shot prediction, which is just, probably just seeing how different the embeddings are and using that as a predictor. But obviously, you could put real data in and try to do better than that. But it's impressive that you can make predictions based just on the output of the model without even training it for the task that you're trying to perform. Okay, so then if we were to train it, How well does it do? Well, we can go back and we can compare it to, for example, SpliceAI, which was specifically designed to just predict these splicing sites, and it actually does about the same if it's trained for that task. I want to mention this briefly. I don't know how widely used this particular model is, but this is a paper where they took the nucleotide transformer that we just talked about, and they put another head on top of it to kind of improve their performance on downstream tasks. And I want to talk about it because that segmentation head is really interesting. It uses an architecture that we're going to see again and again, and that's a UNet architecture. And so UNet is it's a type of CNN that's is often used to like segment different images. So take an image and find the regions of interest, and they're kind of using it to do the same thing in DNA, like take a big DNA sequence and find the areas of interest. And so the UNet, it's basically like a convolutional neural net, but we start with a DNA sequence of of length L. And then it's converting that into a sequence of half that length, and then it's converting that into a sequence of a quarter of that length. And that's generally the structure of a UNet. And these are all convolutional layers. And so we generally end up with more channels and shorter sequences. So it puts it through this bottleneck and it kind of like downscales it into this lower resolution representation, and then it upscales it again back to the original resolution. And it's used for all kinds of things, image segmentation. When we talked about diffusion models, and the neural, this is often the neural net that's used to denoise images, and for example, in diffusion models. And the other key part of the UNet is you can see those lines going horizontal. So these are also those residual or skip connections. We're just taking the output at that layer before it's been upscaled or downscaled, and we're passing it along to the output. So we have the high resolution, the original high resolution information added to the high resolution information that's been upscaled, right? And you can kind of imagine why this would be effective for denoising, right? You're taking a high resolution image, you're collapsing it down and then building it back up. And so you're just going to get the like the basic features. If you go to low res and back to high res, it's going to kind of smooth things out and interpolate smooth lines instead of noisy lines and things like that. Okay. So just some of the predictions coming out of SegmentNT. They're also quite impressive. Here you can see the predictions of splice donor and splice acceptor in different colors. And this model actually predicts introns and exons as well, which is a once you have predictions for splice donors and acceptors, then you need to combine all those predictions together in order to actually predict introns and exons, right? Because if you have two splice donors very close to each other, now you have to figure out like, oh, which is the real one, which is the one that's actually going to be used. So it's a slightly harder problem and potentially more useful problem. So they perform quite well on that. They perform quite well on predicting poly-A sites, enhancers, promoters, like a wide variety of downstream tasks, because the input is based on this like very large pre-trained model. And when they take the when they try to build a predictor that isn't based on an upstream model that isn't pre-trained on a large corpus of DNA data, it doesn't perform nearly as well. So the training that's happening during that mask prediction task is really what's powering like a lot of the prediction coming from the model. Okay, so Hyena is another really interesting architecture, and so the nice thing about this particular DNA model is it's single nucleotide resolution, and it really passes data all along these very large sequences in a way that is computationally a lot less expensive than trying to do it all in a transformer. So transformers are great, but we saw part of the reason for going to a CNN for our handwritten digit instead of just like splaying out all the pixels and letting connecting every pixel to every pixel. Part of the reason for using the convolution is that we didn't want to do that much computation. We didn't want that many parameters in our model, and it's the same logic here. Running a transformer that can look over these enormous long sequences is going to be very difficult. And so the way that they get around it is they use convolutions, but in addition, they have more of the structure from some of these RNNs, these recursive neural nets, where they're taking information and they're allowing the model to gate it, and they're allowing the model to look at the information from one string of convolutions and then learn what parts of that information to pass along. And so they have different stacks of convolutional layers, and then they're gated before they're recombined. And this ends up seems to work quite well and is able to pass information over like great distances in the sequence. But it's only like an L log L operation instead of an L squared operation, where L is the length of the nucleotide sequence. Okay, so this is a figure from that paper, from the Hyena DNA paper, and it's I think it's interesting. So these are different types of genes. And like here's a view using the Teasney algorithm, where we can kind of see what's going on in the latent space and like the embedded space of these different genes. And so the first one is DNABERT, and you can see there's some good separation of different types of genes. The same is true, maybe a little less so. It's hard to tell with nucleotide transformer. But then when we look at the latent space for Hyena DNA, it really looks a lot crisper, and it looks like things that genes of the same function seem to be clustering a little more tightly in more like homogeneous clusters than in the other two. And this is kind of an argument that whatever's going on inside this Hyena-based model, it's creating a representation that might be more informative, less noisy than what's going on even in the transformer-based models. This is a paper that came out pretty recently, and so it's called Evo DNA, and it's another DNA foundation model. And this one is it's building on the Hyena architecture, and it has I think it has a little bit of a different gating structure, although maybe not. I need to go through it. But this is it's using the Hyena architecture with some areas with some attention layers, and so they call it a striped hyena model. And this that they've used it to perform a number of different tasks, and it seems to perform quite well. It does mRNA expression prediction, which is a very hard problem, and seems to do okay. Um, and so that's kind of one of the models to look at. Um, this is I think like a really interesting application that is a little different. So this is a model called Borzoi, and the idea behind this model is to take the human genome and then look at a big corpus, not of DNA sequence data, but of RNA expression data, and then try to predict in 32 base pair windows across the genome, how much coverage of that region do we expect to see in an RNA-seq experiment? And so the structure of the model now should look kind of familiar to you guys. It's pulling together concepts that, like, at this point we've talked about a lot. It's basically a UNet, right? And it's starting out with high resolution DNA, looking at 16 mers of nucleotides. And then it's downscaling that. So it's only looking, it's looking at 32 base pair chunks, and then 64, and then 128. Um, and then once it's in that lowest resolution format, then it goes through transformer blocks. And then the output of those transformer blocks then is upscaled back to not back to the original 16 base pairs, but it's upscaled to 32 base pairs. And of course, it has those residual skip connections. So like all the information from the high resolution uh, convolutions is passed on by adding it, and then they use the output of that last layer uh, uh, to predict the coverage in a RNA-seq experiment. And so what you can see then on the right for this particular gene, EFGR, and here we're looking at RNA-seq from fibroblasts, is like it's pretty good, right? I mean, it's a hard problem, and so the fact that it can reproduce it so faithfully means that like that model has it's learned a lot. It's learned something about chromatin structure. It's learned something about promoters. It's figured out the splicing problem, right? Like it had to have figured out the splicing problem on its own just by training it to predict how much of these things we would expect to see in an RNA-seq experiment. Um, so this is a cool model. If people are interested in playing with like embeddings from DNA from large DNA models. My friend Max Imikov, he made a problem set for another class for me last spring, and he took the final layer embeddings from the Borzoi model for the entire human genome, and that was one of the data sets that we used on a problem set last semester. So it's kind of a big model to run on your own, but we have all the embeddings, and so if people are interested in that for like a final project or something like that, like using that data, let me know and you can have that. Okay, that is it for today, and I will see you guys on Tuesday. I think when is the PSET due, Jared? Is that Tuesday or Thursday? Monday. Okay. Um, and we'll start a new unit on small molecule drugs. I'll talk to you guys about drug development, and then we'll learn some new tools that we're going to use to look at molecules. So see you then. Is tomorrow a mentoring session? Yeah. Yes.